All right, everybody, we're going to go ahead and get started. Thank you, everybody, for being here. It is nice to see so many faces in the room. My name is Cinnamon Moffett. I'm the research program manager here at the Hatfield Marine Science Center located in Newport, Oregon. This is a hybrid event, so we have folks that are also online. Thanks, everybody online, for being here. If you have any technical issues, Roseanne is in the room. Hi, Roseanne. And she will help you. So just put any of those questions into the chat and she will work through those with you. Also, for folks online, we do have your cameras, mics, and screen shares turned off. But if you have questions for today's presenter, if you put them into the chat, we'll read them out to the room at the end of today's talk. For folks in the room, we've got a couple of different groups here joining us. So the AQS program is here. So thank you so much for being in the room. We do ask that if you haven't already, that you sign the little sign-in sheet as you come in the door. The only reason I need that is I have to justify that I am not buying cookies for myself. I'm buying cookies for you, so I need a head count. So please sign in. That would be great. For folks that are online or in the room that are taking this for OSU credit, Dr. Kate Stafford is also in the room and she is monitoring your questions. So know she is watching you. So please participate with us as you see fit. A couple of quick announcements before we get started. I just wanted to remind folks that our next Science on Tap is next week. It is October 4th when Katarina Alden is going to come and talk to us about how the discovery of a whale nursery transformed an entire community. So that is on the 4th, starting at 6. We will do a social hour starting at 5. So hope you can join us then. She will also come back the following day on the 5th and be a part of our seminar series where she dives in a little bit deeper and talks about what she did wrong while she was doing this study. So I'm excited to have her come and share with us. Her home base is in Mexico, so we're excited to have her join us next week. But while everybody is in the room is for today's speaker. So let me just do a little introduction. Many of you already know today's speaker, but Matt Hawkyard is an OSU alum who did most of his graduate work and his postdoc work here at Hatfield. He then left us kind of quietly in the middle of COVID and snuck out in 2021 and moved his family across country and took a position in the Maine Aquaculture Research Institute, where he does animal nutrition and food technology, focusing on early stage marine fishes. And so he has a lot to share with us today. So I'm just going to stop talking and hand this off to Matt.
Yeah, thank you, Cinnamon. Thank you, everyone, for coming today. It's really my pleasure to be back here and in front of so many friends and colleagues. And you know, as Cinnamon mentioned, 2 years ago, we made the big journey across the country. There's the sign that says Boston, 3,000 some odd miles away. So we literally just took Highway 20 almost all the way to Boston and then jumped up to Maine once we got close enough. So it's been a sight unseen. We had never been to Maine, and it turns out to be a really beautiful place, and it's been a really wonderful place to go work. And so to me, I just was excited to come back and share what I've been up to for the last 2 years. I can't talk about everything, but I'm going to kind of make a little bit of an attempt to talk about at least some of my favorite projects. So so thank you. Yeah, I'm going to start with talking about, advances. And then, okay, I'll do it like this. Okay. So I'm going to start with talking a little bit about why I think that why we should be investing in aquaculture. And then I'll give a little brief overview of what's going on with Maine's finfish aquaculture industry. Then I'm going to kind of talk about 3 of my favorite projects. The first one, using insect meals as an alternative to fish meal. Then I'm going to talk quite a bit about the developing larval diets for early stage, particularly marine finfish, work that's continuing from the work that was started off working with Dr. Langdon and during my graduate work and has evolved through the years. And then finally, if I get time, I'll talk a little bit about lumpfish because they're so charismatic and it's a fun project. So starting off with why should we invest in aquaculture? And okay. So, you know, we've got 8 billion people. We just crested 8 billion people on the planet by the most recent census. And during our lifetimes, it's very probable that a lot of us are going to experience quite a few more than that, with upward estimates of reaching 10 billion people, in somewhere in the 2050, 2100 range. So that's going to be stressing, all of our food systems. We really have to think about, how are we going to produce food to feed all of these people with raising quality of life happening, in developing nations. So it's not just about producing enough food, but, providing a quality of life and nutritious food for those people. And it really is going to take all of the tools that we've got as, food producers. So this is a paper I really like. This was done at the Bren School of Economics by Froelich et al. And they basically looked at, let's look at some potential ways to grow our food systems in the future. So one way to do that would be, let's just expand all of our agricultural and aquaculture systems proportionally into the future. That would be the BAU model, the business as usual model, or we could really invest disproportionately in aquaculture, whether it be mixed, so that's freshwater and marine, or just marine aquaculture. And the short of this slide, the 1-minute version or less, is that it takes less crop usage and it takes less land usage to be able to expand aquaculture, to grow a similar amount of food, to be able to feed people at these these future projections of what we're going to need to do in, say, 2050. The reason why that is because a lot of that model growth is happening in finfish, and finfish are very efficient at converting food into food, right? So for the data people, that's up there in terms of FCR, that's a food conversion ratio. It's the lowest among animals. And then there's all these other conversion efficiencies. But the important thing when you think about fish is that they are neutrally buoyant, right? So they're not spending a lot of energy supporting their own weight. And they're poikilotherms, so they're adjusting their body temperature to the water around them, which means they're not burning a lot of energy for thermoregulation. And so those two things combined really set them aside from a lot of our agricultural animals that we try to raise and make them very efficient at turning food into edible protein. So sometimes I think we get wrapped into this dichotomy of, capture fisheries versus aquaculture. I think that's false. There is a role for all of these. We're going to need all of these tools into the future. But when you really look at production fisheries in the light blue, this is FAO or SOFIA, FAO data from the last SOFIA report. And, the fisheries just haven't been putting out more fish, globally speaking, since the late 1980s, early 1990s. It's really been pretty leveled out since then, and some doing quite well and some fisheries not doing quite so well. But really, all of our excess need for seafood has been met by aquaculture over that time. You see a dramatic increase in aquaculture over the same time period. And now about half of the seafood available to people is coming from aquaculture. Well, that's mostly happening overseas. We're not responsible for that development, I'm sorry to say, in North America, particularly in the United States. We're in a huge seafood trade deficit. So in 2020, we had a $17 billion trade deficit, right? So most of the seafood that we're eating at the seafood counter is coming from overseas. So why is that a problem? Well, that's a problem because, one, you've got a lot of stuff being shipped to us. There's carbon footprint associated with that, but also just thinking from a food security standpoint. We're all starting to get more aware of the fragility of our global supply chains. Things like the war in Ukraine have been very disruptive for wheat production and for commodities, really leading to a lot of the inflation we're seeing today. We're also seeing things like, the COVID virus really changed our perception of our supply chains and our food security. And I think this kind of thinking has the Department of Homeland Security actually commissioned a report that was just released in the last year that really was looking at the threats to our national food security. And what are those leading threats? And it's actually a very scary report because some of this has to do with like intervention from foreign adversaries and such. But one of the top 6 recommendations was actually increasing domestic aquaculture production as a means of stabilizing and securing our own seafood security in the United States. And then this isn't aquaculture or fisheries or agriculture. It's just something we all have to deal with. I stole this slide from Chris some years ago, but just the idea that climate change is going to be affecting how all of our food systems are able to produce food into the future, right? There's a big question mark on how we're going to be able to do business and there's going to certainly need to be some climate adaptability built into, you know, how we think about this stuff. And I'm going to talk a lot about finfish and finfish aquaculture. That's my role at the Aquaculture Research Institute, is a finfish nutritionist. And I guess one of the questions would be, why focus on finfish? And, you know, American consumers are, well, our number one thing is shrimp. So it's twice as much shrimp consumed by Americans compared to the second most eaten seafood. But really, most of the seafood that we eat by large is our finfish, right? So if we really want to change what consumers have available to them and produce that domestically, we really have to think about how are we going to make more finfish available to the consumer. Okay, so how about a little bit about Maine's aquaculture industry? Well, Maine actually has a lot of similarities when compared to the Pacific Northwest, and you kind of combine Oregon and Washington together. We're growing scallops. There's a pretty sizable oyster industry there. One of the largest kelp producers in the U.S., lower 50 is there. We've got multiple blue mussel growers, and this is not the, Dr. Damian Brady gave a seminar to the Hatfields 6 months, maybe a year ago now, but he covered a lot of this. But just to highlight, you know, it's quite a diverse portfolio of aquaculture. But, you know, from a finfish perspective, traditionally, Maine has had net pen aquaculture in the state for a couple decades now. Is largely under the umbrella of a single company, and it's almost all salmon aquaculture. This is one of their marine sites. But there's also this interest in moving to land-based, not moving to, but including and adding land-based aquaculture into the portfolio of Maine aquaculture. And there's a lot of kind of excitement and some movement in this area. So these are 4, there's multiple companies involved, but here are 4 that I just wanted to highlight that have a kind of a high chance of success or are already have a strong foundation. So American Unagi, they're taking wild harvested glass eels and then growing them up to adults for the sushi market. And they actually have a built out facility in Walboro, Maine. Kingfish Company, they have an incubator, a small pilot facility on the University of Maine campus on one of our satellite campuses. It's a business incubator. But they are actually going to be building a facility in Jonesport, Maine, and they're in kind of the last, getting through the last barriers to get that moving. But it looks very promising that they'll be started this next year, breaking ground. Nordic Aqua Farms, that's the one you probably, if you're up in the news at all about Maine RAS, recirculating aquaculture systems, these land-based systems, they've been in the press quite a bit. They're going to be in Belfast potentially, but they have run into various hurdles that, you know, have slowed things down a lot. And then finally, this Katahdin Salmon, they just proposed this facility 6 months ago, and kind of probably due to their location and the embrace by the local community, they may be first up and running. And so this gives you an idea of kind of all of this, not those sites and then more, the landscape of Maine aquaculture geographically speaking. So the orange circles I'm showing, those are marine net pen sites. The blue stars, those are actually salmon hatcheries to support largely right now, the net pen operations. The slashed blue star is an under construction lumpfish hatchery that they'll be doing for biocontrol, and I do talk about that at the very end. And then all the red stars, solid red means they're already operational land-based systems, and then the striped ones are these proposed and in various stages of moving forward. And then finally, you know, on the University of Maine, we have money, although I just got bad news. It's a little bit less than we thought. But we're going to be building the Sustainable Aquaculture Workforce Innovation Center. So this is going to be a land-based aquaculture facility for research purposes and workforce development that'll actually be on our campus. And it's going to be really exciting, where we've actually hired the construction managers and the architects, and it was kind of promised when I took the job that this would be happening, or at least a carrot's kind of dangled, and it actually is happening now. So that's pretty exciting. So yeah, let's get some of the research. I wanted to start by talking about one of these projects where we have looked at insect meals as an alternative to fish meal. So, you know, why would we do that? Well, you know, when you look at particularly marine aquaculture, offshore, you know, near shore and offshore aquaculture, those systems, feed costs represent the #1 variable cost for those operations. So it can represent almost 60% in some cases. All those pallets, each one of those bags contain feed that's going to go out to this site, and that's one morning feed, one daily ration that they've brought out on forklifts, and they're going to barge out to that site, and split among many net pens. But you just get an idea that the investment that has, and about 1/5 of that food there is made out of fish meal. So that fish meal is coming from, right now, it largely comes from wild captured fisheries, reduction fisheries, places like Peruvian anchovies, menhaden in the Gulf of Mexico, these sorts of reduction fisheries. And what we've seen over the last 20-30 years is a decline in the availability of that reduction fisheries for fish meal. This is FAO data. And largely that's as those fisheries have moved towards sustainable certifications and had pressure to reduce and not overfish, which is a good thing, of course, but it has trimmed the availability down. Plus you have annual variation in those fisheries. El Nino is probably going to throw another wrench in the fish meal production facility for this year. And then you've got the orange line is actually the utilization of fisheries byproducts for fish meal usage. And so more and more processors are looking at ways of taking their byproducts, the trimmings that are coming. So the Pacific Seafoods, the hake factory, there's actually a fish meal processing plant just upriver. Last I knew, it wasn't yet able to provide animal feed fish meal, but it requires substantial tooling to be able to do that. But I'm sure that's on their radar in the future. And that's why there's that uptick. There's getting to be more financial incentive for these processors to look at that. Okay, so, and more aquaculture is taking the lion's share of fish meal. It used to be kind of shared between the poultry and the pig industry. And now really in these days, the aquaculture folks are just willing to pay more for that resource. You can think about fish meal usage in another way, and that's in fish in, fish out ratios, right? So this is really thinking about it in more an environmental context and in an efficiency context. Does it make sense to catch 4 pounds of wild fish to grow 1 pound of salmon? And that's been a big argument. And I think that was actually really brought to the forefront by Rosalind Naylor in 2000. It really created a lot of uproar in some ways, but I think some very good thinking about how we were using these wild fishery resources to feed aquaculture. But two things have happened. One, that brought that attention to the light, and that has really driven, I think, the industry to do better. There's also an economic incentive, which I'll get to next. But basically, what by all metrics, where they think we're at now, by best estimates, is about 1 pound of fish in for every pound of fish that we're growing. That's for salmon, and it's pretty similar for trout. It'd be less, globally speaking, when you start including things like carps and catfish. But we can do better. And this is the economic incentive, right? That's soybean meal on the bottom. This is cost per ton of soybean versus the cost per ton of fish meal over a 20, 30 year almost 40 year time series. But the point is that it's cheaper if you can find alternatives to fish meal, and that cost keeps going up and is highly variable and based on fisheries dynamics. And then finally, just talking about insect meal specifically, people have been thinking about a lot of different alternatives. And actually, when you look at fish food, it's made out of all kinds of stuff. There's even a feather meal that you can see that's usually some sort of fermented feather meal. And that's an animal feeds broadly speaking, but there's a whole wide range of very interesting ingredients in them. But when we look at these alternatives for fish meal, two that really stand out are the single cell proteins, so bacterial and yeasts. When you look at the red vertical line, that's the line in which they think is the upper. This is based on a meta-analysis. That would be the upper inclusion level without having a negative impact on the feed efficiencies of the animal, converting feed into protein. And so both single-celled and insects show the highest ability to have very high inclusion rates without impeding the growth or the efficiencies of our target animals, the fish. So we were this was contract research that we did from a French company that is growing mealworms and then converting that mealworm into mealworm meal. And so basically they take food waste and then they incubate the mealworms on that food waste, and then they dry that down into a powder. So if you hear the term meal, that means the product has been converted into a powder, so it's more easily, handled and through extrusion and all these processes. So we did a digestibility study first, and then a growth study, looking at this insect meal. So I got to go out to Bozeman, Montana. This is my colleague Michael Hapasian. We were actually hired at the same time as both to kind of build out this fish nutrition program at the University of Maine. And so we flew out to Bozeman working on this, and making fish feed is not an easy task. So if any of you have ordered fish feed for feeding animals, or particularly research scale fish feed, there's a lot of work that went into that. There's the formulation, there's the weighing of the ingredients, you got to mix the ingredients. That thing on the right, that's a hammer mill. So you got to grind everything down into a pulverized, easily handleable powder. And then that's before you even get to the extrusion process. So then the powder gets dropped. That's called the mash. The mash is being dropped into here. That's a twin screw extruder. So there's two long screws in the center of that it's for safety reasons, you can't see the die, but there's then some blades that are cutting that food up into pellets. So I don't remember if this was our growth diet or our digestibility diet, but then you have to dry the food and cool the food. And then with salmon diets, we actually have a very high lipid content salmon diet. So we actually put the oil in through a vacuum coating process, and that's that piece of equipment on the right. So we made for a digestibility study, the way this is done in fish is that first you make a reference diet. There's two methods, but I'm going to talk about the 70-30 method. So first you make a reference diet, and then you make two or however many. In this case, we had two test diets where we wanted to test defatted mealworm and whole mealworm, and you mix them 30% of the test ingredient with 70% of this reference basal mix. This is the basal mix. You don't need to know it, but it is standard for our program and also for the USDA program. So we have a standardized basal mix so that we can compare between studies. And the way this is kind of done is this is my best attempt at trying to summarize this in like 1 minute or less. But you basically look at the reference diet and how well that's digested. We put yttrium oxide, which is a rare earth metal, into the feed to use as a marker. But you look at how much you compare the feed with the feces. And basically what we're doing is seeing that in this case, I'm showing that the reference diet, maybe it was 50% digested, but the reference diet was 70% of that test diet. So then we assume that the 70% was 50% digested to be 35%, right? So just throw a ton of percents at you real quick. And then the remaining fraction can be attributed to the digestibility of your test ingredient. That's my short version. So, but the way that actually looks in practice is that you've got a bunch of tanks. You can have the fish in there. They get anesthetized. You pull those fish out. I think this is me hand stripping a salmon, doing my best to get the feces in the cup. So you probably didn't expect some poop research going on in this talk today, but that's a big part of this. Then those samples are all then freeze dried, and then we do nutrient analyses of the various nutrients in the feed, and then the analysis of that inert marker, and through some fancy math, you can see how much of that test ingredient was digested. So when it comes to the insect meal, about 90% it was about 90% digestible. That's very good. And then I know there's a bunch of numbers up there, but I'd bring it down to fish meal. There's a couple fish meals there. They're usually on the order of 83 to 94% is herring. Menhaden around 86% digestible. So we're actually doing the insect meal is quite digestible, and that's very good. So it means that they can use more of that product, and it also means there's fewer waste materials left, which can be important in some systems. Okay, so we also performed a growth study. And so in the growth study, we did this in a small scale system with juveniles, and we had 4 experimental diets. So a fish meal based diet. And when I say that, that means 20% of that diet was made out of fish meal, and then 80% was made out of other ingredients, things like fish oil and vegetable oil and whole wheat gluten, the whole suite of different practical ingredients that we'd find commonly in North America. And then we replaced half of the fish meal with a defatted mealworm, or all of the fish meal with defatted mealworm. And then we also had this one that was a product that was not defatted. So as we call it, whole mealworm meal. And that we did that at a 50% replacement rate. And just getting straight to the, this is a 12-week growth study. So at the end of those 12 weeks, we pulled the fish out and found that they actually grew statistically similar among all 4 treatments. And so that's a good thing. This is one of those studies that it's great because you can publish it if there's a difference, and you can publish it if there's not. So very safe. But one of the interesting findings here was that we saw the IgM and IgT. These are immunoglobulins. And this is RNA expression. So they were upregulated. Both these immunoglobulins were upregulated in the 50%, both of the defatted mealworm meal diets. And so, this is one of those things we don't really know if that's a good thing or a bad thing. If we had followed it up with a challenge trial, it may be that it acts as an immunostimulant, and that might confer some additional protection if done in conjunction with vaccines, that sort of thing. This could be good, or it might be indicating that there is some sort of kind of pro-inflammatory or at least immunogenic compound in that meal. So that is going to require further investigation. We also looked at the gut microflora, and so this was working in collaboration with some folks at Florida Atlantic University. But basically, what you eat affects the microbiome in your gut. So if you go on vacation and you eat a bunch of stuff that you're not used to eating and your stomach feels upset, part of that is the microbiome of your gut responding to this change. And that's kind of what we saw. We didn't see it in terms of Shannon diversity, which means kind of the number of organisms present in a given system, but rather we did see it in the types of organisms, and that showed up in this principal component analysis, where we did see the kind of relative abundance show up in the principal components. And that's shown better here in this bar chart, where this is actually breaking it down into species of the microbiome and family and genus, and basically you see that they're represented in there's changes in the proportional representation of those different organisms. And so again, we don't really know what's best. We assume fish meal is the kind of the gold standard, so we assume that's the best. But we know there's a change. It's hard to then kind of do much more than arm wave about those differences, but certainly it does influence gut microbiome. And then finally, this is a question that's kind of a new one that you would add to a study like this, because we're thinking about using these materials in land-based systems. The producers are really interested in the feces and how well they hold together in the systems. Because if they break apart a lot, then you can get a very highly turbid system, and that can create gill irritation. And it's also much harder to filter out small materials when compared to big materials from a cost standpoint. And so they don't want the feces breaking down into a bunch of suspended solids. So we looked at that with a laser diffraction particle analyzer, and this is after agitating the feces with mechanically, and these are feces that have been stripped straight from the animals at the end of the study, not that they weren't collected from the system. And the short of it is we didn't see any difference between the defatted mealworm and the fish meal diets, or the feces originating from that. We did see a non-statistical, but this trend where the whole mealworm meals feces actually showed a higher proportion by volume of particles in that small size range. So there's a little bit of a caution there for the company moving forward with that particular product in land-based systems. I think it was really fun. I just thought it'd be a fun one to highlight because it's a little different than stuff I ever did here at the Hatfield Center, and that is being done typically here. So, and then our take homes is that these are the insect meal that we worked with, the mealworm meal, was highly digestible. The salmon grew quite well on it. We did see this upregulation of immunoglobulins that warrants some further investigation. And then just to thank the folks for Insect for having funded that. And it's a French company, but they do plan on building a North American plant and are looking into that now. So that's kind of exciting. Then I'm going to shift gears quite a bit. This is a big shift, so get ready for it. But this is jumping into developing larval diets to improve hatchery efficiency. So this is work that Chris Lang and I have worked on for years. It's something that I've been able to just bring with me to the University of Maine and start a whole new lab making micro diets, and have had to set up a whole basically mini processing facility in-house. It's been very challenging and very rewarding at the same time. But the things that we're thinking about when we're feeding very early staged fish are things like retention of water soluble nutrients. So I'll go into that a bit more. Digestibility, so making sure that the animals have access to the things that you're encapsulating. And then things like acceptability and buoyancy. So making sure the animals will eat those particles, that they're in the water column long enough to have access to them. And I didn't put a bullet point, but up there, there's a lot of other considerations, but nutritionally adequate would be another important one. So, what's the problem with early staged diets is they're very small, right? So a small fish needs small food. And what you have, if you look at a juvenile diets are like an early stage grow out diet might be in a 3 millimeter range. But a larval diet, a 0.3 millimeter or 300 micron particle would be a pretty standard kind of first feed commercial diet. And when you look at the surface area to volume ratio of those two different particles, meaning the surface area being the kind of the area where the things would leach from, versus the volume of the particle, which is that storage capacity, or what you would hope would be the protected portion of that particle. Your surface area to volume ratio changes significantly going down an order of magnitude in scale. The surface area to volume goes up an order of magnitude, going from a surface area to volume ratio of 2 to 20. And that means your propensity for leaching goes way up at these smaller scales. And then to illustrate this was work I did actually here at the Hatfield Center with Fred Conte when he came up to visit. We put some sodium fluorescein in a commercial type diet that was actually done by Rick Barrows, a collaborator. And we sprinkled those diets into a tank, and so it has this green fluorescent dye. And this is what you would see if you were just feeding, your fish, but under a black light, this is what's actually happening to those water soluble compounds. And I've gotten a lot of mileage out of these slides, but it's so nice to see this, in terms of just watching the leaching occur in real time. So this is after 2 minutes. You can see the diets are starting to settle in on the bottom. You can also see particles sinking down and the entrails of that. And this is 30 minutes of those diets sitting at the bottom of the tank. And that's not uncommon in larval systems. They don't eat much of the food. They're not that effective eaters yet. And so, this is a great way to grow bacteria if that's what you want to do. But there's this trade-off. We can encapsulate a lot of these compounds, but then they have to be digestible. So you could put them in a titanium shell, but that's not going to give the fish any access to the contents, the payload of those particles. So we have to kind of take that into account as we improve some efficiencies, others might. There are some trade-offs involved. And then I wanted to highlight acceptability and buoyancy because it's something that we've had to work on quite a bit. And you're working with these early stage animals that are going, from live feeds, so rotifers, Artemia are commonly cultured live feeds that we feed in place of copepods and captive systems. But as the fish age, we can wean them onto artificial diets, these micro diets, the dry feed on the top. When I was a graduate student here, we worked a lot on using liposomes to enrich live feeds, and then feeding those that's a northern rock sole on the right there. But we found that they were very effective for that. These liposomes are microparticles that were developed for pharmaceutical purposes. And we've adapted them for use in aquaculture. And the green dye, they don't actually glow like that. We've put green fluorescent dyes in them so you can visualize them, but they're made out of, these are soy-based envelopes that we can put a variety of different water-soluble nutrients into. And they worked really well for live feeds. So these are rotifers enriched with the liposomes, then Artemia on the bottom enriched with these liposomes that are fluorescent. And they made fish grow, and develop better. So the fish had access to the contents of those liposomes. And we see that in, the taurine liposome enriched rotifer fed, I know it's a mouthful, but fish on the bottom left really show that, the fish were growing really well as a result of that. So the next step was then, well, could we take those liposomes and put them into a larger carrier particle so the fish could eat them directly? So that's what I'm going to spend the rest of my time talking about, is not just liposome complex particles, but those and commercial type microdiets. So on the left are particles where we actually took the liposomes and embedded them into these larger gelatin alginate particles. And then the water soluble payload that we want to deliver is inside the liposomes. And then on the right is a commercial type microdiet. So this would be something you'd buy off the shelf today. A product like HMSC is the one I commonly cite. You take all the ingredients that you want to deliver to the animals and you grind them up. They go through an extrusion process basically, and they would be all in that matrix on the right. Well, when we've done leaching trials with taurine, a water-soluble nutrient for fish, we found that the complex particles retained a very high fraction of that payload of the taurine over a 1 hour period, whereas the commercial type microdiets lost a very significant fraction of that water-soluble nutrient over, within minutes, but certainly after 30 minutes, they basically had, 15 minutes, they have negligible amounts of taurine in them. So that was pretty exciting. We then used that to look at how could we modify the flavor of these particles to change ingestion rates. And the short of it here was, if we put something like glycine in these liposomes and very simple empty particles, we actually saw that the fish ate much higher levels than if the particles were just filled with empty kind of saltwater liposomes. That was kind of early research, but then this current project is to then say, well, can we deliver complete nutrition with these complex particles with the liposomes embedded to deliver a kind of key water-soluble nutrients? So we know fish larvae do better if they're fed, pre-hydrolyzed protein that's already kind of been pre-digested a bit. It's much more easy for the fish to further digest and then assimilate that type of protein than something that's completely intact in, say, a fish meal or a squid meal. So that sounds great. But this has been a huge amount of work to get to that video on the right, where we're actually now producing the liposome-based complex particles with the spray system at the scale needed to run larval feeding studies has been pretty challenging and required, as I mentioned, kind of building this whole processing line. But we've gotten there, and that's pretty exciting. So we're actually able to make 6 kilos of that recently, and we've also looked at means of modifying the buoyancy. So when we first offered these to fish, they kind of sank like a rock. But now we have 2 methods of adjusting the buoyancy of these particles, including using gas forming agents, kind of create little air pockets in the particles, and then adding hollow silica microspheres to the center, inclusion particles that can modify the buoyancy. So that's pretty cool. Now we are able to make kind of adjust the particles to a more near neutral buoyancy. And we've done some work looking at how much liposome we can what percent of the particle could be made up of liposomes. And I won't go into that too much, but what we do know is the more liposomes we put in, the higher lipid contents we see result. And that protein goes up as well, but there's this point where we don't want any more lipid in the particles. So that kind of sets some of our constraints as to what degree of water-soluble payload we can deliver. And then I'm really excited about this. This is equipment that we recently purchased through a National Sea Grant that is on the left is a microextruder, and on the right is a spherinizer. And so this is how we think Odihime, a very common commercial microdiet, commercially purchased microdiet, is made. It results in those spheres on the right, but this allows us to now make commercial diets that we can formulate just like our complex particles and use in parallel studies. And so we have this kind of commercial control with the same formulation as our experimental particles. The other thing is I've got a whole project now to just work on this spheronization process so that we can actually look at better ways of retaining water-soluble nutrients, making them more acceptable, and then starting to tailor them for some of the key species that we want to grow here in North America, like Seriola, California yellowtail, tailor them for lumpfish. And so anyways, this is a very exciting kind of addition to the portfolio. And anyways, we just got this stuff, so it's still shiny and new. But the other thing we're doing is taking these microdiets, and we want to use ingredients and formulations that people can interpret and reproduce elsewhere. So one of the challenging things is if you use a commercial diet as a control treatment, you don't really know what's in it, and it's really hard to make progress nutritionally in nutritional experiments, certainly, but even other experiments when you're kind of working with this black box. So NOAA, folks from NOAA and USDA, long before me, developed this open formula concept and published this and really have encouraged it. But those other folks are retiring and kind of moving out of the spotlight. And so, I felt like this was a great opportunity to grab onto that concept and then apply it to, our complex particles and our commercial type micro diets. And we can share this. We can get, hopefully other labs get on board with this, and we have kind of more standardization across and make it more industry and broader progress throughout collaborative efforts. Okay, so we made a bunch of diets recently and ran a trial at Hubfield Research Institute, but we had these commercial type diets on the left. I've shortened it down to MEM. Those are microextruded marumerized particles, the liposome complex particles, notoheme. And the nice thing is, through the use of these buoyancy modifying particles, we're actually really able to adjust the buoyancy of those particles and get them to where they were, much slower sinking. So you actually want a very low number in this case. That means they're sinking more slowly. Then the commercial diet otahime, and then our MEM particles actually sink very similar to otahime, which also is a very good sign because we thought they were also going to sink faster than otahime. So this is great. We then offered in the fish, and this is where this is new information, hot off the press. So feel free to leave right now if you want. But basically, the downside is that the fish just didn't eat the complex particles at the rate that we had expected them to do. But we did, there was a lot of success in here, just getting to this scale, and we learned a lot through just going through the process. You got to do something first once, you have to do it the first time at least one time, right? Or however that saying goes. So anyways, I felt I'm pretty proud of just having gotten this all done. But now there's a lot of work to figure out what went wrong here. What didn't the fish like? Was it the actual physical property, how the mouthfeel, so to speak, of the particle? Was it something about the preservative that we used to preserve the complex particles, or was it lacking something that they needed as a cue to get them to eat? So this is our next trial to run. We're going to be running an acceptability trial. We did follow this through in a growth trial, and this is a mixed bag of our complex particles. Fish that weren't eating didn't grow very well, so no surprise there. But room to improve, which is great. What I'm really excited about is the fact that our microextruded marumerized diets and the commercially purchased otahime diet fed fish all grew at the same rate, which we did not expect at all. We thought otahime, being kind of the best diet out there right now, would outperform our open formula diets, and it would take some research to get equal, and then we need to do more research to get better than. That's kind of our longer term goal. So we're starting off on really good footing with that technology, with that process. And then right now are just have a lot of kind of exciting things to move forward with that. I'm here to give a research report for Western Regional Aquaculture Center project and working with folks here. So I'd be remiss if I didn't mention this part. We're also taking the liposome-based complex particles and putting them into putting vaccines into these particles. So this is a collaborative project with University of Idaho, Oregon State, and University of Maine. And what you see on the left are the complex particles that have whole cell bacteria in them. So those are GFP, fluorescently stained bacteria, and on the right, the liposomes in the particle. And we can put that all into the same particle. And we've actually gotten to the point where we've challenged the trout and sablefish with an Aeromonas salmonicida, a live pathogenic bacteria, after having been vaccinated with an Aeromonas A-cell vaccine using different vaccination strategies, neither an injection strategy or a bath or immersion vaccination, or a couple of different control options. And where we're at with that right now is that the oral vaccine is doing about as good as a bath vaccine, which is actually pretty good. They do use bath vaccines for commercial purposes, and there may be some benefits to having an oral vaccine as opposed to an immersion. But this is a very challenging one as well. We never pick the easy projects, I guess. But the industry has been wanting an oral vaccine for some of these for vaccination purposes for decades, and has run into a lot of challenges. So this is actually not a bad result. With sablefish, not quite as responsive, but they did do better than the non-vaccinated fish. And now we're looking at this oral vaccination platform in combination with other, as a pre or post vaccination booster to have a kind of a near-term takeaway for industry. Okay, so anyways, lots there, but our next steps are to improve acceptability of liposome-based complex particles so that we can get back to the growth studies, to investigate methods to improve these commercial type micro diets. So lots of work to do there. That we're in the first year of that project. And then also the vaccine stuff, looking at kind of other applications for this oral vaccine. So it may be, you know, we may just switch to a different pathogen because Aeromonas salmonacida, its infection pathway is not through the gut. So switching to a pathogen that reflects that infection pathway, we may have a lot more success. So we've got some ideas on how to move forward with that technology as well. Lots of funding, lots of partners, some of you in this room. So thank you for all of that. And then finally, just I think I got just enough to kind of squeeze in my last project. It's just fun. So, but there's no research here. This is just a little, I guess, tidbit for the afternoon. But lumpfish, they're a big problem in the salmon industry right now. And this is pan-Atlantic, is that the sea lice. So sea lice are related to copepods. They're an ectoparasite, and they wreak havoc on the salmon industry. And my friend who loaned me this slide said, don't hover on it too long. That's what they look like. And in Maine, they're not allowed. There's a chemical pesticide that they have used in Canadian waters that has not proven to be. It's getting less efficacious over time as the copepods are developing resistance. So that's a big concern in Canada. In the U.S., we haven't allowed the use of these pesticides. Well, at least in Maine, they're not allowed to use that. I don't think in the state of Washington they ever were either. And so currently in Maine, they have to keep the levels of those copepods down, or they would have to cull that entire pen. And so the only way to do that right now is through this mechanical operation. So we went out to this marine site when they were treating, and this is a big water slide that the fish get sent down, and they're spray bars spraying the copepods off with heated seawater. And then so they're pumped up, go down the water slide, have a jolly good time, and then go back into the net pen. But this is, you know, it's stressful for the animals. It's a huge operational cost for the company because once they start this, they can't stop. They'll start in the spring as soon as if they start seeing these infections, and then once it starts, they're kind of locked in to doing that for the rest of the year. And so this takes obviously a giant barge, a bunch of people to operate that barge. There's losses of fish in some occasions. So this is not their long-term goal. So to get away from that, they're building a new lumpfish hatchery in Maine that's going to be located up in Eastport, and so and their pilot program is located in Franklin on the UMaine campus, and also in partnership with the USDA, who we work very closely with. But anyways, these are lumpfish. So lumpfish are cleaner fish. Those are the kind of young of the year over there on the left, juvenile lumpfish. And then you see a couple pictures of adults or near adults there on the right. And they're very effective. You put the lumpfish in the systems at about 1 lumpfish per 10 salmon. And the Norwegians have had really good success with this, and the Canadians are having good success with this. So now we're just adapting this for Maine. But as such, we have to come up with our own protocols and such to make it applicable to our own hatcheries. And I get to go on a boat ride and collect lumpfish. And we got these seaweed mats. You find them, they associate with these floating seaweed mats. And you grab the seaweeds and you pull it out and you kind of beat it on the side of the cooler, and the fish drop out into the water. And then we drove around, and if you're driving around, and this was Frenchman's Bay, which is like by Acadia National Park, which is a great excuse to go for a boat ride. And we went past like 3 lighthouses on the day on the water. So anyways, this is just fun. And then finally, this is we're doing an NRAC. This is a Northeastern Regional Aquaculture Center project to develop some feeding rates for the hatcheries, and we're going to build that into a lumpfish hatchery handbook. So anyways, there's some work that has to be done there. And with that, thank you for your attention. Appreciate everyone coming. So thank you.
Thank you so very much. There was a lot there. So we're going to bounce back and forth between folks online and folks in the room for questions. Are there any questions online, Roseanne?
Yes, there are. First of all, Bill Han Shoemaker says hello, and he asks this question. Does mealworm fish food contain sufficient omega-3 fatty acids for salmon culture?
Yeah. Hi, Bill. Yeah, so no, so in this is a formulated feed. So the nice thing about a formulated feed is that it's using like 20 different ingredients in it. And this one maybe didn't have quite that many. But what we can do is you can use different ingredients to meet the needs, the different nutritional requirements of that animal. So mealworm meal on its own right does not have things like DHA or EPA in it in any significant quantity to meet the needs of salmon. So that does mean that we have to also find a source of those oils to put in the feed. And I didn't go into that because I kind of felt like maybe I talked about enough today. But fish oil is another big concern for the industry. So it's actually going to be relatively easy to replace fish protein because there's nothing that special about protein. You just need one that's digestible by the fish and doesn't have any anti-nutritional effects like causing inflammation or anything like that. But the fish oil challenge is going to be a really big one because there aren't a lot of terrestrial alternatives, particularly for DHA and EPA specifically. So yeah, good question. Thanks, Bill.
All right, questions in the room.
Thanks. Just a quick question. It sort of follows up on Bill's. You've been talking about growth, and so growing a fish out is great, but like, what effect did the change in these meals have on the actual quality of the fish? It's ultimately like you want to take that to market. Is there someone that's looking at the effect of these different meals on what the actual like taste and all those things, and longevity of the fish on the shelf and things like that?
Yeah, so that wasn't part of our study, but that's certainly something that is a consideration when looking at alternatives to fish meal for protein sources. So when you look at things like people have used protein extracted from corn, for example, and put that into fish food. And one of the key things that can lead to from a consumer preference standpoint is discoloration of the fillet, where you get more of like a yellowish fillet as opposed to that nice pink fillet that you might otherwise want. So yeah, certainly like ingredient choice can have a big impact on the resulting fillet quality and consumer preference. And there are studies that certainly aim to answer those questions. And we just didn't, wasn't within the scope of that particular project, but something that could, be built into that. I haven't seen anything that has shown that consumers are off put by insect meals being fed to fish. And in fact, a lot of fish just eat. I mean, you look at trout, insects are a major mainstay of wild trout diet. So that one's not that crazy. But when you look at, soy and corn and some of I think particularly when you look at terrestrial protein sources, then, fillet quality becomes much more important. Sure. Yeah. Well, okay. So just a little tidbit about that. One big problem with land-based aquaculture right now is this stuff called geosmin. There's also MIB. These are off-flavor compounds. And so it turns out if you're growing fish in an enclosed system, you can get, we think it's due to the microbiome of the system, not of the animal's gut, but that you get these off-flavor compounds that can give it kind of a dirtier taste. So commonly tilapia will have that. Like if you've had tilapia and you thought, oh, that's an earthier taste, that's geosmin in there. And so what they're seeing, that's another big problem. Like sea lice being a huge one for the offshore people, but off-flavor compounds is a big concern for the RAS, the recirculating aquaculture system producers. And the anecdote, well, I don't want to talk. I'll stop there.
All right. Questions online. Go ahead, Roseanne.
Thank you for the amazing summary report. Marine net pens seem increasingly efficient and effective in marine systems, sort of like feedlots that have been an evolution from hatcheries. East Coast lobster production seems to have transcended the feedlot to become free range, but dependent on the bait for increasing fractions of their diet. Since you're already making fish adapt to artificial diets, are there prospects of free range finfish aquaculture advancing in the same direction? Striped bass, for example, beg for food in Woods Hole. How are they different than sheep or cows? And he's asking because the pens seem to be increasingly expensive and vulnerable.
Yeah, okay, that's a big question. Yeah, so parallels with the lobster industry, that's a good one. Partly one thing, this is my own speculation, not speaking, I guess, as a researcher, but just as a speculative person. I think that one thing that'll happen is we'll move towards land-based systems, and then people say, hey, actually, those net pens, there's very little energy use there, and they're living more in their natural waters, and that in the long term, not now, people here, you're not ready for this, but 40 years from now, when your children's children. I think there will be this move back into the marine environment. We'll start looking at offshore aquaculture and even nearshore aquaculture systems differently than we do now, and maybe design them in a way that we're building parallels to free range terrestrial agriculture, which aren't necessarily the free range that you envision, but larger pens and higher animal welfare. And animal welfare is a key concern for industry right now, and there's a lot of discussion around animal welfare and how to do that from all aspects, and what does that mean? And there's some really interesting kind of guiding documents coming out about animal welfare right now. So very good question. I don't know if I answered it or just danced around it, but that's what I said. So,
All right, questions in the room.
Other than the salmon lice and the copepods there, what are your main concerns for ecto or endoparasites?
sorry, other than
the lice that you're talking about with the lumpsucker, with the
oh yeah, sea lice. Yeah, are there other ectoparasites of concern? I'm a nutritionist, I suppose. So I do work with a lot of aquatic animal health folks, and I haven't heard of them being particularly concerned about any other that are wreaking havoc, at least with Atlantic salmon or other net pen farmed animals, with the exception that, geez, in the Pacific, with some marine finfish, they get an ectoparasite, and if I recall right, it's a trematode, but I totally could be wrong about that. It's not. Does anyone else know? And it is really challenging for them to treat, and it's a small, like salmon aquaculture is so far advanced of where we're at with marine finfish, and so this is like a new problem, and they don't have the same kind of infrastructure, broader industry and research investment to treat that, and so that is a kind of a big area of concern for a select number of offshore growers in the Pacific. And it is, I believe it's a trematode, but don't hold me to that, but it is kind of more of like a worm-like ectoparasite. So.
All right, we've got time for one question online and one question in the room. So Roseanne, can you pick one?
Oh, and there are potential for cleaner fish on the West Coast too. The lumpfish is not a good one out there, but there are, I think there's 11 or so species of lumpfish in the Pacific, but they're mostly littler, and I don't know if they would make a good cleaner fish. But I know that the Shiner surf perch, that the folks here have long used them as cleaner fish in the exhibits. So that's one thing that the Canadians are working on. So there might be some application for yet another cleaner fish for Pacific, ectoparasites, just to tag onto there. Yeah.
Okay, Roseanne, go
Sorry.
ahead.
Maybe, do I still have one question left?
You've got two. We're gonna do them quick. Go ahead,
Okay,
Roseanne.
does using insect meal introduce any potential diseases for aquaculture?
Oh yeah. I don't believe that we've got, so, we haven't seen any sort of indication that insect meal specifically would be challenging for disease pathogens that fish could get. But that is something that to be concerned about, depending on how the food is processed and where those, how that's handled from like whether it be fish meal or insect meal or anything. So, but no, I don't think there's any evidence that insect meals themselves are transmitting pathogens through the prepared formulated feeds. Yeah.
On the last one.
Last
First off, great talk. Thank you for coming to talk with us, and thanks for sharing the lump sucker pictures. They're so cute.
Yeah.
My question is about, well, when I think of insect proteins, I think of like cricket flour that you can find in grocery stores. And I was curious how like mealworm meal compares to cricket meal, and if there's any like pros and cons for why you went with mealworms, and if that maybe it competes with our demand because it seems that we like cricket flour or it's growing. I don't know. Thank you.
Yeah, I know. I think broadly, like insect meal, there's a lot of potential there for not just for fish meal, for fish diets, but certainly in livestock diets and in human consumption. You know, I mean, they're more prevalent in some diets overseas than they are here in the U.S. But I've seen them in corner stores from time to time, chocolate covered crickets, for example. But I don't know anything about crickets, honestly, and the cricket meal. That's not something I looked at. We have looked at black soldier fly. So those are that's the kind of the competing insect meal available, or that's becoming available, and that they're actually commercial producers working on now. So black soldier fly being kind of a ubiquitous fly that they lay their eggs into food waste, and then their larvae then consume that waste, and they can consume the larvae. And so they're different. They have their digestibilities are going to be a little bit different. Their composition a little bit different. Their levels of chitin, that's a big one. That's their exoskeleton would be made of chitin, and so those levels may be different. And but the other thing is the black soldier fly are much more lipid rich, and we know that they have some ability to modulate the fats that they eat. So they kind of like store the fats that they eat more than the mealworms that are doing more bioconversion. So it'd be more from the oil standpoint, I think, where you'd see the big difference between those two. But I can't speak to crickets, sorry.
All right. I'm going to wrap us up there because we're at end of time, but I will say that there are more questions in the room than we can get to, and definitely more questions online. But if you just go to whatever platform you use and you look up Matt Hawkyard and UMaine, it comes up. I think the first one is information on you. So you can Google yourself, but you can find his information. You can contact him with any questions that you might have. But if we can give one more round of applause for our speaker today. All right, everybody. Thank you so much for being here. I hope you see you next week for both our Science on Tap and our seminar. For folks that came for credit, make sure you do what you need to do. And we're so glad that you were here. Thanks, everybody.